This lecture will discuss the principles of antimicrobial chemotherapy and identify plausible targets to treat infection. The learning outcomes are as follows. 1. To explain the principles of chemotherapy and the process of appropriate antibacterial drug selection. 2. Using drug examples, describe the major pathways and processes disrupted by antibacterial drugs. 3. Describe how bacterial mutations can lead to resistance to antibacterial drugs. In selecting antibacterial agents, we first of all need to identify the species and its sensitivity using laboratory methods, such as those shown in the diagram on the right. The picture shows an agar plate with bacteria growing on it, as well as different antibiotic discs showing distinct zones of inhibition. This can help identify the most efficacious drug for a specific infection. In the absence of such data, selection will be influenced by patient history, location of infection, gram stain, identifying a gram-negative or gram-positive species as is shown in the diagram on the bottom of the slide, and finally site of infection. This slide gives a broad overview of the species that cause different types of bacterial infections as well as the anatomical locations they will most likely affect. For example, we see species that cause respiratory tract and GI infections, as well as STIs and meningitis. As a matter of principle, chemotherapeutic drugs should be toxic to invading organisms, but non-toxic to humans. They should have selective toxicity, most cases relative rather than absolute. It is important to consider the potential effects on the microbiota in the gastrointestinal tracts, lungs, and vaginal tracts. Drugs should target unique genetic or biochemical pathways to pathogens. Drugs should be selective to target protein isoforms unique to the pathogen without affecting human forms. And drugs should target metabolic requirements that are specific to the pathogen. Some important points to consider when selecting a drug include its inherent toxicity. Many are among the least toxic drugs, while others, such as chloramphenicol, are less specific. It is also important to consider patient factors like their immune system, because the host must ultimately eliminate the invading organism. Renal dysfunction, as we will see, can play a role in selection, as accumulation can occur in some when normally eliminated in others. Hepatic dysfunction also can affect those drugs or their metabolites that concentrate in the liver. Age is a consideration, as renal and hepatic elimination processes are often poorly developed in newborns or they malfunction in the elderly. Finally, in pregnancy, all drugs cross the placenta. Adverse effects are rare, except for the tetracyclines and aminoglycosides. For nursing infants, they also can receive antibacterial agents in the breast milk. It's important to clarify and classify some terms. Antibacterial drugs interfere with the growth of microbes within a host. They may be synthetic or derived from a living organism. An antibiotic is a substance produced by a microbe that in small amounts inhibits another microbe. These are a subclass of antibacterial drug. Bacteriostatic is a term that means the drug stops growth or proliferation but does not kill the bacteria, and host defense is required to defeat the infection. Bactericidal, on the other hand, means it kills the bacteria. These terms are better illustrated by the graph shown on this slide, which shows the log growth of bacteria on the y-axis and the number of generations following division on the x-axis. It is clear from the graph that growth can be affected by varying degrees of inhibition, from no effect to those that are rapidly bactericidal. It is also important to consider the number of species of organisms affected by the drug. Narrow spectrum drugs act only on a single or a limited group of microorganisms, for example isoniazid against mycobacteria, whereas extended spectrum drugs are active against gram-negative bacteria such as ampicillin, and broad spectrum can affect a wide variety of microbial species. These drugs can drastically alter normal bacterial flora and can precipitate a superinfection, for example with candida, normally kept in check by other microorganisms with tetracycline and chloramphenicol. Chemotherapy can target different types of reactions in bacterial cells. These can be classified as follows. Class 1 reactions are those that use glucose and other carbon sources are used to produce ATP and simple molecules. These tend not to be promising targets because humans and bacteria use similar mechanisms to obtain energy from glucose. And even if glucose is blocked, other sources of carbon can be used by bacteria. Class 2 reactions are pathways utilising energy and class 1 compounds to make smaller molecules such as nucleotides. Agents that interfere with folate synthesis can be used, for example. Class 3 reactions are pathways that convert small molecules into macromolecules such as proteins, nucleic acids and peptidoglycans. Agents that interfere with protein synthesis or with RNA polymerase or DNA gyrase, for example. Agents that target formed cell structures such as cell membranes would be found under this category. Shown in the diagram on this slide is a bacterial cell with the reactions that are targeted by the most commonly used antibacterial drugs. These reactions include nucleic acid synthesis, cell wall synthesis, folate synthesis, RNA transcription, protein synthesis, and cell wall disruptors. We will discuss all of these in more detail over the next couple of lectures. The table on this slide shows these same families of drugs, their specific target, whether they are bacteriostatic or bactericidal, and some specific examples of the group. Also shown is a timeline indicating the periods of discovery of antibacterials. Most drugs we still use came from the golden era of discovery, with some others being modifications of these drugs. Modern discovery is dealing a lot with overcoming resistance and investigation of narrow spectrum drugs. The first major target to consider is the bacterial cell wall. Bacterial cell walls contain peptidoglycan, which is not in eukaryotic cells. In gram-negative bacteria, it is a single layer thick, but in gram-positive bacteria, it can be up to 40 layers thick. Each layer consists of multiple backbones of alternating N-acetylglucosamine or NAG and N-acetylmuramic acid or NAM, as shown in the diagram on the top right. NAM residues are cross-linked together via an interpeptide bridge, as shown in the diagram on the bottom right. Peptidoglycan synthesis involves a number of different enzymes and transporters in bacteria. Shown in this diagram are some of the potential targets for this process, as well as the drugs currently used to inhibit synthesis, including the beta-lactam drugs, vancomycin, bacitracin, and cycloserine. In addition to having a cell wall, bacterial cells also have a cell membrane. Plasma membrane of bacterial cells is similar to that in eukaryotes, in that it has a phospholipid bilayer in which proteins are embedded. As is shown in the diagram on the right, this is present in both gram-positive and negative cells. Certain antibacterial drugs act as detergents by disrupting the phospholipid components of the membrane and kill the cells. Polymyxins are cationic peptide antibiotics that contain both hydrophobic and hydrophilic groups. Inhibiting the synthesis of nucleotides is a class 2 reaction. Folate is required for DNA synthesis in bacteria, and some antibacterials interfere with this pathway, such as the sulfonamides and trimethoprim. Altering the base pairing properties of the template and intercalate with DNA, increasing the distance between the base pairs and causing a frame shift mutation. Direct effects on DNA itself include using alkylating agents that can affect the formation of covalent bonds with bases in DNA and prevent their replication. Drugs that follow this particular mechanism, such as bleomycin, tend to be used in cancer chemotherapy. DNA replication is a key target for antimicrobials, including antibacterials and antivirals. As shown in the diagram, a new DNA strand is synthesized with the involvement of a number of different enzymes and proteins. This firstly involves unwinding and unzipping DNA, and DNA polymerase adds complementary nucleotides in a semi-conservative fashion, forming a leading strand and a lagging strand. This can be interfered with in bacterial cells. DNA is a drug target, therefore, and a number of enzymes surrounding it can be inhibited, including the DNA polymerase enzyme, DNA gyrase or topoisomerase, as well as RNA polymerase that's involved in transcription. Protein translation is central to any living cell survival. It occurs at the ribosome and involves the transcribed mRNA combining with two subunits of the ribosome. Individual transfer RNA molecules carrying unique amino acids arrive at the ribosome and interact via an anticodon specific for the 3 nucleotide codon on the mRNA molecule. It then deposits its amino acid to a growing polypeptide chain via peptide bond formation. This can be a significant target for antibacterials due to differences between human and bacterial ribosomes. Eukaryotic and prokaryotic ribosomes are different, and this provides the basis for selective antimicrobial action. The bacterial ribosome consists of a 50S and a 30S subunit to give a 70S complete ribosome. The eukaryotic ribosome consists of a 60S and a 40S subunit to give an 80S complete ribosome, as shown in the diagram on the top right. To initiate bacterial translation, mRNA must bind to the 30S subunit first and then bind to the 50S. The ribosome has 3 binding sites for tRNA, termed E, P, and A, as shown in the diagram on the bottom right. Antibacterial drug resistance is quite a big problem, and we have many examples of this occurring, most notably MRSA or methicillin-resistant Staphylococcus aureus. In the 1940s, we had the first case of penicillin resistance, and MRSA began to appear in the 1960s. In the EU alone, in excess of 3 million people develop an infection arising from hospital-based infections, resulting in approximately 50,000 deaths per annum. 1 in 10 patients, it's estimated, entering a European hospital will develop an infection caused by drug-resistant microbes, such as hospital superbugs like MRSA. At the moment, it's estimated that 50% of strains of bacteria like staphylococci are resistant to many of the first-line drugs such as penicillin, methicillin, tetracycline, and erythromycin, and TB infections caused by Mycobacterium tuberculosis are often resistant to the first-line drugs like isoniazid and rifampicin. The diagram on the slide here shows the amount of antibiotic consumption which is thought to contribute to some of this resistance that has arisen over many years. This is a heavily researched area now, and consumption can be measured in defined daily dose, which is the usual dose per day for an adult. As can be seen from the map of Europe, Ireland is still in the mid-range compared to some of its neighbours for this. Selection of genetic variants within the population is what causes drug resistance. Resistance is heritable via certain genes called OR or resistance genes. They can often carry mutations in a drug binding site which can lower the affinity of the interaction with the drug, as shown in the diagram on the top right. They can also sometimes have genes encoding for enzymes that degrade antibacterial agents, rendering them ineffective. Finally, they can carry genes coding for efflux pumps, which actively remove drugs from the cytoplasm as soon as they enter, as is shown in the diagram also. It is important to remember that bacteria have higher rates of mutation to us, estimated that 1 in every 1 by 10 to the power of 7 cells is mutated. This can often be carried on extrachromosomal plasmids that replicate independently, known as R plasmids, as shown on the diagram on the bottom right. These resistant genes can be carried on mobile genetic elements, on transposons, and recombine in new cells, as is shown on the diagram below. They can be transferred via transformation, transduction, and conjugation, as is shown in the diagram at the bottom of the slide. Transformation involves the release of DNA and involve it being taken up by a recipient cell. Bacterial transduction involves the transfer by bacteriophage viruses that insert DNA sequences containing resistance genes in the chromosomal plasmids. Finally, bacterial conjugation involves the direct interaction of two neighbouring cells and the transfer of copied plasmids.